Now, part two of our culturing of bacteria and microbes in the laboratory setting. Now, as we just got done defining in part A, or discussing in part A, and defining the synthetic or the defined media versus complex. One you know everything about, the second one you kind of have an idea, but you really don't. So you're just making it super rich. When it comes to purpose, well, different augers, different broths serve different purposes. The general purpose, brain heart infusion, tryptase soy auger. As I said in part A, this is going to be so rich. This one is so rich, has so much there. You could live on it. It's not going to taste good. It's not going to give you all the calories you need for the day, but it is so rich that most microbes, most bacteria can live on this and do very, very well. But it turns out some microbes need a little extra. That's where the enriched media comes in. Enriched media means that you're going to add blood or serum to a general purpose media. Here on the right, you see this red and you see this dark brown augers. The red, blood auger. Tryptase soy auger is liquid when it's heated, and as it cools, it solidifies. As it's cooling, once it hits a certain point where it's cool but not solid yet, you pour in some sheep's blood. The sheep's blood, it's going to have red blood cells in it. It's going to have serum in it. Means there's going to be some form of growth factor present. A growth factor that certain microbes are going to require. These are going to be pathogens that generally infect humans, animals, and lyses red blood cells, lyses animal cells to get at certain nutrients that are not going to be present in a soy digest, or you're not going to get from just boiling brain and hearts. The brown, the dark brown, well, it's roughly the same as the blood auger, but the blood is added at a higher, so when the auger is at a higher temperature, so that the heat lyses the red blood cells. Blood auger is red because of red blood cells. Chocolate auger, which is what that brown one is dark brown because all of the lysed and oxidized red blood cell proteins. Blood auger, you use it so that you can determine which microbes are able to lyse. You see this halo around the dots, the colonies, the bacterial growth. That's where the bacteria have been able to lyse the red blood cells to get at the proteins and things inside, the growth factors. Whereas here, this bacteria needs those growth factors inside the red blood cells, but it lacks the enzymes to be able to lyse them itself. Now, when it comes to being able to culture microbes, a lot of the times you're not going to have, somebody's not going to just give you a tube, give you a swab that there's only one type of bacteria on it. The majority of the times, there's either going to be more than one, or you're unsure if there's more than one. So what you can use are media, agar broths, that will allow you to select. So for certain bacteria to grow, and others will struggle and not be able to grow due to bile salts, too much salt being there, not enough of certain amino acids, different things like that. if you look at the table 2.3 on the right there. Couple of different types of media. The McConkey agar is one that's used quite often. It has within it it's a complex. It has bile. We're not sure what's in the bile. We just know there's going to be bile salts and things like that. And then crystal violet. The bile salts are going to interfere with gram-positive bacterial growth. The crystal violet is going to get caught up in the cell wall of gram-positive bacteria, making it harder for them to grow. So the gram-negative intestinal bacteria are going to grow very well on McConkey agar. All other bacteria will struggle and probably much not be able to grow. You look down there at the bottom. Sauerbrads agar has a pH of 5.6. That's a highly acidic pH. Most microbes don't like that. Bacteria can't grow with that. But this is really good for fungi because it mimics the decomposing nature of plants and animals where some fungi are going to want to grow. Then there's differential media. Differential is going to allow different things to grow, but due to some of the components within the agar, different bacterial species are going to turn different colors, as you see there on the right. Some reds, some yellows, some off-whites, depending upon the bacterial species. Selective lets you select who can and cannot grow. Differential, whatever does grow, it lets you tell the differences. Here's a figure from the textbook, kind of give you an idea. You take a mixed sample, you spread it around on a general purpose agar. Anything and everything grows. Lots of the different sizes of the dots supposed to represent different species of bacteria. But over here on the selective media, only one type can grow. You do the same thing. You take the same mixed sample and you put it on differential. Most things can grow, but notice they take on different colors based on what dyes, what compounds are in the agar. Differential media. We talked about the blood agar has red blood cells in it. Lets you tell the differences between which bacteria have the ability to lyse red blood cells and get at those growth factors and which one can't. Mannitol salt has mannitol, a sugar mannitol in it. Very few bacteria can utilize this sugar. The Staphylococcus genus primarily is the only one of medical importance that can. Has phenol red in it. Other microbes can grow there on this agar, but only Staphylococcus, Staph aureus, Staphylococcus epidermidis, and those can utilize the mannitol, ferment it, change the pH. Phenol red goes from a nice red to yellow as the pH becomes more acidic. McConkey agar has the sugar lactose. Again, not very many microbes have the genes necessary to utilize the sugar lactose. Those that can will ferment the lactose, changing neutral red from red to a yellow. Or I'm sorry, going from clear to a red. It's hard sometimes keeping all these straight in your head. Urea broth. Which microbes can break down urea? break down urea to ammonia, and the ammonia will then be utilized for the making of different amino acids and other differentials that allow you to tell the differences between the different bacteria growing there. And then there's, the catch-all. There's miscellaneous. Miscellaneous such as the reducing medium. It gets rid of any oxygen present. So this is going to be for bacteria that like to grow in a unoxygenated environment. Carbohydrate fermentation media, as you see here on the right, starts as a red, and if the bacteria can utilize the one sugar present, the one carbon source present, it goes from red to yellow. This upside down tube, you can just barely make out inside each of these, known as a Durham tube. So if the bacteria can utilize the sugar, usually it's dextrose, lactose, and sucrose, one of those sugars will be in the tube. If it can use it, boom, red to yellow. If it can use it, but it's using it in a fermentation process, which means a gas bubble, gases will be produced, the upside down Durham tube will capture the gases. You'll see a bubble. So it's, yes, it uses the sugar, yes, it produces gas. Now, inoculation, incubation, and finally, isolation. You can't sit there and run tests. You can't sit there and identify a mixture of bacteria. If it's more than one species present, you can't do it. So you have to be able to isolate. Isolation is separating one species from any other that could be present. Best way to do it is to start off with one bacterial cell. Turns out bacteria divide. 1 becomes 2, 2 becomes 4, 8, 16, 32. You go through 32 rounds of that, you have a few million bacteria. You start off with one bacterial cell on an agar plate. You wait 24 hours or so, you'll have what's called a colony. 1,000,000 clones of that one original bacterial cell. I can't do any tests on a mixture of bacteria. I can't do any tests on one bacteria. But 1,000,000 clones that are genetically identical, genetically, metabolically identical to the original cell, that I can do something with. You look at this figure from the textbook. A mixture of bacteria. You figure out a way to spread them out, isolate one cocci, one bacilli, one sphere, one rod. You separate them out on an agar plate. You get one bacteria will suddenly become millions of its clones overnight. That's a dot, as you can see right here, a colony that you can see, a colony that you can pick up, remove from the agar, and you can run tests on. Now your textbook points out there's multiple ways of isolating. And if we were in lab, we would do this first one, called the streak plate method. You take a metal loop, you pick up the one colony, or you dip it into the mixed culture, and then you smear that around in one area of an agar plate. You then put that loop into a Bunsen burner or into an incinerator to sterilize it. Now it's clean. You're going to take and you're going to gently brush from where you just smeared across. Pick up, lay down, pull, smear, smear, smear. Incinerate again, sterilize, and do from here over, here over, here over. So as you see on this plate here, the lines where they went this way, were sterilized, then went this way, sterilized, then went that way. Notice red, white, and lots of yellow. I can't do anything in this mix. So that original broth that had this mix of bacteria, I really can't do much with that. But you get over here, an individual red colony, individual white colony, individual yellow. That's millions and millions of the red, the white, the yellow I can pick and I can do stuff with. There's also this pour plate method. You take that loop, you transfer over into one tube, you mix it into some liquid, you pour it on top of an agar plate. You take a little bit of it, you transfer it over and transfer it over. So you're diluting high concentration, lower, lowest. And as you see here, not as many on this final plate. Individual white, individual yellow, individual red. Streak plate uses only one plate. It may not always work. Most of the times it does, but you see here, you start off with a lot of bacteria. Good chance you can end with individual colonies. This pour method or this spread plate method, which is quite similar to the pour method, going to utilize quite a few, 5, 6, 7 plates, depending upon just how concentrated your bacteria are and just how much you want to dilute it. You can do it as few as 3, as many as 6. And then you have to look through until you find the plate that's shown right here or shown right here where you have individual red, yellow, and somewhere in there is white. Red, yellow, white. All nice, all by themselves.